Good morning. Again, I apologize. I'm going to be speaking English and not Portuguese. I have not learned it from yesterday, so still English. Also, I see a lot of different faces from yesterday. Yesterday, I spent the first half hour explaining our laboratory and what we do and how things work in Canada a little bit. So I didn't want to repeat that this morning, and I'll just go on and talk specifically about a second project. Do you want to chair? There's a lot of extra chairs. Another difference from yesterday to today is yesterday I talked about Doppler optical coherence tomography, and we did a lot of work on that. So we did the basic physics, optics, engineering, then preclinical testing, and then clinical testing, the whole continuum. And so it's a fairly mature program. We have a lot of students, a lot of postdocs, a lot of clinical people working on this. Whereas this is a little more early stage. So this is still more kind of at the physics, biophysics, engineering stage. We don't have any clinical results yet because we don't know if it's going to work or not. So it's a bit of a difference from yesterday. Also, as I mentioned yesterday, if you have questions or comments or don't understand the science, the physics, or my English, please interrupt and stop. We have lots of time, so we don't need to wait till the end. Okay, so we're going to talk about polarized light. And the basic idea is very simple. So if you have optical light that has polarization and you shine it on tissue, is there any information that comes back that still has some polarization information? And if the answer is yes, and we'll show you that the answer is yes, what can you use it for? So the basic idea is you take light and you shine it into tissue. It scatters many, many times. So it zigzags, multiple scattering. So if you shine red light, you see this big diffuse glow. That's multiple scattering. And some of the light escapes, comes out. Is there any polarization left? And if the answer is yes, what can you use it for? So you launch in polarized light, linear, circular, elliptical, any other states of polarization, and some of it is retained upon return. And you measure these polarization properties of the light that comes out. So why is this interesting, useful in biomedicine? Well, there's several reasons, and I list some of them here. So the ability to use polarized light to see through tissue, to see through a very multiply scattering medium for optical imaging. That's one possibility. Another thing that we are very interested in is to quantify tissue. It's a complicated word. Anisotropy means different properties in different directions. So for example, if you have muscle, the optical properties in this direction are different from this direction. And that displays itself or manifests itself as so-called birefringence. Again, very long words, but maybe similar in Portuguese, birefringence, something. A third one, and I'll give you an idea of where this is very important in biomedicine. A third interesting application, probably the most important one, is trying to quantify glucose or glucosa blood sugar level in diabetic patients. As you probably know, most diabetics have to inject insulin and have to regulate how much they eat and how much they exercise. But the proper injection and regulation of that depends on knowing how much glucose there is in the blood at any one time. And right now, there is no way to know that unless you pinprick your finger, draw blood, and then you can find out to within 10% what your glucose level is. So what the Diabetic Association, for example, American Diabetic Association recommends that diabetics have to do this 4 times a day, 4 to 5 times a day. And the problem is it's not just today and next Thursday and 2 weeks from now, but it's every single day. And it's for children, for medium-sized, for adults. So it's a big, big problem. People continuously do this. So the problem is most people don't do it. Most diabetics do it once a day, once every second day, but very few actually do that, do the 4 times a day every day. So it's a huge, huge problem in clinical medicine to develop non-invasive methods for determining blood sugar level or body glucose levels. And there's been optical approaches, there's been RF impedance approaches, there's been ultrasound approaches. And the market and the clinical need is so big that there's tens of companies continuously working on this, continuously coming up with products, continuously marketing products, and all of them don't work. It's just a very, very difficult problem because the normal physiological blood sugar level is 1 gram in a liter. So this is half a liter, so half a sugar cube in that, and it's not transparent, but it's like a glass of milk, like latte. So the question is, if you have milk, no milk here, and it has sugar in it, how can you tell if the milk is sweet or just non-sweet? And so you're looking for 1 gram in a liter, that's in blood. But blood is 10% of the body. So it's 1 part in 1,000 that you're looking for. And you're looking to see about 20 or 30 or 40% change in that very small level. So that's a very small change in a very small signal. But physiologically and medically, that's a big deal. 40% change in your blood glucose level, that's, especially if it goes down, that's very, very dangerous. So there's a big commercial and clinical need to develop methods of non-invasive glucose monitoring for diabetic patients and for other glucose analytes. So we think that polarized light may have a role to play in it. And the reason we think so is because in transparent media like this, no scattering, no multiple scattering, it's very easy to use polarized light to determine whether this is plain water or sweet water. Does anybody know how you can do that with polarized light to know if there's sugar in this water? Any idea? Right, optical rotation or optical activity. Glucose is one of these structurally asymmetric molecules, so-called chiral. Chiral means from hand, so left hand and right hand. They're similar but different, and the images don't look the same. So as a result of this asymmetry, if you shine in linearly polarized light that goes through here, it comes out linearly polarized, but the plane is rotated. And by noticing how much rotation there is and knowing how thick this was, you can determine the concentration of glucose. And that is routinely done in biochemistry, in laboratories. But it requires clear media. So if I have a little sugar and water, no problem. But now in tissue, imagine the same thing, but now I had milk or latte. So I shine in light, there's nothing coming through in a straight line. You just get this big diffuse glow. So can you use polarized light to see if that milk or if that scattering tissue has any sugar in it and glucose? And if yes, can you know how much, and can you see differences in those signals? So that's kind of one of the rationales, if you want, for trying to use polarized light in tissue for glucose monitoring. There's also some very interesting physics in this, and fundamental biophysics of disordered media, or random media, or turbulent media. So I'm sure most of you know what this is, but polarized light is just basically the direction of the electric or electromagnetic field perpendicular to the direction of propagation. So I'm sure most of you have seen polarized sunglasses, and they actually polarize this way. So the reflection of the water surface is blocked because that polarization mostly in the horizontal direction. So basically polarization is just direction of the electric field in perpendicular to where the light is going. So if I shine light at you, it's what this is doing, this way, this way, this way, this way, or randomly. So that's unpolarized light. So direction is, so these are the common linearly polarized light, circularly polarized light, this way or this way, left or right, or elliptically, which is just a superposition of these two, gives you elliptically polarized light. So in some ways, this is the most common way. This is a special case of a round ellipse, and this is a special case of a very elongated ellipse. And this is when all of this happens too quickly. So when you look at it's white light like this, which is random, all of this exists, but it's happening too quickly, so you get unpolarized light. So the math or the models we use for dealing this, again, I'm sure this is a bit of a review to some of the people. You may have heard about Stokes vectors, Jones calculus, Muller calculus. They're basically descriptions of how you do the mathematics of polarized light. So I'll come back to this a little later. So this is called something to describe the light. The properties of the light itself are basically four measurable quantities. This is the common one. This is the intensity. So just how much light there is, how many watts, how many milliwatts, how many joules, whatever units you use. These two describe the linear polarization, how elongated is the ellipse. And this is describing the circular polarization. This can be positive, right, or negative, left. So if these are all 0, then the light is unpolarized. So you can have 1 watt, 0, 0, 0, means it's random. Or you can have 1, 0, 0, 1. That means the intensity is 1 and the polarization is fully right circular, fully left circular, or any combination thereof. So that's how you describe the light. How do you actually describe what happens when the light propagates through tissue or through water or through a lens or through a mirror or any combination thereof? Is you use something called the Muller Matrix, which actually has a lot of elements. So 4 by 4, 16 elements. A lot of, this is kind of like the transfer function. Those of you who study engineering would know what that means. So it's what the medium does to the input and how it generates output. So this is how you describe tissue, how you describe polarizers, how you describe analyzers, how you describe modulators, etc. And this is also how you can describe tissue, if you know how to do that. And so the basic math is fairly simple. If you know the polarization state of your incident light, say it's fully circularly polarized or fully linearly polarized, you know this vector, which has 4 entries in it. And you're generally interested in either this or in this. So what you can do is you can measure this with analyzers and polarizers and try to work backwards and determine that. Or if you know by knowing ahead of time or by model what this is, what it is for polarizers, for analyzers, for modulators, and maybe for tissue, if you can make intelligent guesses as to what these entries are for tissue that you're studying, then you can study the output light and use that to characterize what's happening in the tissue and how sweet milk will differ from non-sweet milk, for example. So this actually looks very simple, but can get...
only an absorbed
Sorry?
A medium that just absorbs light.
Yeah, so that's going to manifest itself in certain entries here that will decrease the output intensity. So the I will go down very significantly because you would have real parts as opposed to imaginary parts here, so the light would absorb. So that's easy to handle. What's a little more tricky is those of you who know matrix mathematics would recall that in matrices or in linear algebra, A times B is not equal to B times A. So the order is very important. The effects don't commute. So if you have a medium that's a retarder or a wave plate or quarter wave plate, that's easy. That's one effect. But if you have tissue that can be scattering, that can have optical activity due to glucose, that can be birefringent because it'll have different directions properties. How do you put all that together in one matrix? There's no easy way to do that, because you can't say that it scatters first, then it optically rotates, and then it does the retardance or birefringence, because you don't know that. It does everything simultaneously. But ABC is not equal to BCA, is not equal to CBA. The order of matrix multiplication, especially if the matrices are complex, matters. If the matrices are very simple, like 1, 1, 1, 1, everything else is 0, then of course the order does not matter. But that's a special case. So in general, this looks deceptively simple, but it's not that trivial to do when you actually get, and I'll show you how we deal with it. So that's the math. What are the uses so far of polarized light in medicine up to now? So there's been a lot of work, actually. Steve Jacques and a couple of other groups, Judith Morant in the States, and several others, Rox Anderson, have used polarization to basically select or to gate which photons they want to look at, which depth of tissue. So you can eliminate surface reflection, for example, by using cross polarizers. So if you want to see subsurface skin details, the epidermis, the dermis, and you're not interested in what's happening in the stratum corneum, which is the very top layer of the skin, you can simply view cross polarizers and illuminate that specular reflection. Conversely, if you want to illuminate the stuff that is scattered lots at the bottom, you can use this type of techniques where you can, this is a normal image, and you can do a polarization image to look at the low scattered photons that are just below the surface. So illuminate all the depolarized background deeper. So this is some work that was published in 2002. A lot of interesting work has been done on polarization in terms of sizing of the particles, trying to determine how big or how small they are and what shapes they are and how different they are from circles or from spheres. And that, a lot of work has actually been done in looking at clouds and planetary atmospheres by using polarized light and polarization changes on remote sensing to determine that. And people like Michael Feld and Judith Moran have used this to basically determine if the cells are getting bigger or smaller. And if the cells are getting bigger, or sometimes the nuclei are getting bigger, that's a sign of either cancerous progression or apoptosis, etc. So you can use some of these polarization tricks to get an idea of the distribution of sizes and shapes of scattering particles. It's a little tricky, but it can be done. Some work has appeared on what I was mentioning before, to looking at the different properties of tissue in different directions.
Excuse me, so
Absolutely.
that's not in vivo, right?
That is in vivo, actually. Yeah. Michael Feld and his group and this guy Vadim Beckman have done it in the esophagus, to looking at the epithelial lining of the esophageal wall to see if there's early disease progression.
But don't you have to pass the light through the tissue so you can see it?
They look at singly scattered or doubly scattered light. So they look at retroreflection, generally in the backwards direction, and they do the polarization gating to eliminate the deep tissues. And they look at the size distribution of the epithelial layer, the first 50 microns or so, the first 2 or 3 layers of cells. Very, very clever work, actually. Very clever work. So this is what I was mentioning before, looking at polarization changes as a function of tissue disorganization in different directions. And that's very important. For example, I'll show you some examples when in stem cell research and regenerative medicine. For example, if you have a heart attack or an infarct and stem cells are injected, right now it's kind of a guess as to what happens. There's some clinical evidence that that's a good thing, but nobody really knows. So if we can quantify how the tissue, how the scar tissue is remodeling, and whether it's forming kind of anisotropic heart muscle versus a more disorganized matrix, that's very useful information. So we'll get back to that as well. And a lot of tissue, especially collagen and muscle fibers and elastin, are very different in different directions, anisotropic. Unlike, for example, fat or breast tissue, which is much more isotropic, the same in all directions. And this is the one that we'll concentrate a little more on. I guess these two, but this one is specific, which is you looking at this optical activity, the ability of the glucose to change the linear polarization state. And does that hold in a tissue medium, in a turbid medium? We know it holds in a transparent medium. That's really of limited use in biomedicine. So we'll see if that works. So just to concentrate on glucose, what people have done to date, which has been a lot of work simply because the problem is so important and so difficult, and therefore no one succeeded yet, is to look at the absorption changes in the near infrared. Glucose has no visible absorption spectra features, but in the near infrared and the mid infrared, it does have some. Very weak, because the concentration is 1 part in 1,000. So for every 1 molecule, you have, say, 1,000 water molecules, which is a much stronger absorber in the infrared. So there's a lot of confounding factors, but people have looked at absorption near infrared spectroscopy and changed the intensity, changed the absorption spectrum as a function of glucose, as it changes from 5 millimolar to 7 millimolar to 10 millimolar. In other words, from 1 gram in a liter of blood to 1.5 grams in a liter of blood or something like that. People have also looked, I don't know if you guys are familiar with Raman scattering, which is another vibrational absorption spectroscopy, a little more sensitive, a little more specific, but the signals are much, much weaker than either fluorescence or absorption. So this, in principle, is a much better, more sensitive fingerprinting method, but in practice, it's not so easy, just because the signal is maybe 1,000,000 times weaker than the absorption signal. So we're going to look at using polarization and optical activity changes due to the polarization, due to the polarized light nature, polarization changes induced by glucose. Another important effect that glucose induces is that when it's dissolved in a medium, say in water or in serum or in blood, it actually raises the refractive index of that medium. So if this is invisible, 1.33 is the N, the real part of the refractive index for water. If I add, say, 1 molar glucose, which is a lot of sugar, much more than we have in our body, but say 10 grams, or not 10 grams, say 1 gram to this amount, the index is going to be 1.35 or 1.36. So it's a fairly significant change. You can quantify that change. And what that means is that the scattering properties of tissue change as glucose is up or down in the body. So people have used absorption and scattering spectroscopy to quantify that change, to look at the change in the scattering coefficient as an indirect method to see how much glucose there is. So it actually does two things. There's optical activity, absorption changes, and refractive index matching, which changes the scattering coefficient, changes the path length, changes where the light goes, etc. So polarization specifically, which we'll be looking at, is affected, if you want, or sensitive to both of these, because this changes the path length, and there changes, therefore, the how much light is polarized or depolarized upon scattering, and also induces some optical activity. Question? No? Okay. By the way, if anyone has any questions, please go ahead.
Looks like the basic problem of all these techniques is to distinguish what comes from glucose
and what comes from 50 other things. Absolutely.
For instance, optical activity you just mentioned before, then you have contribution from glucose, but also protein.
Absolutely.
And,
Absolutely.
how do you distinguish that?
Wait, yeah. But I mean, the simple answer is you use so-called signal analysis, like partial least squares, chemometric methods, loading vectors. You're absolutely right. It's not the only thing that changes the refractive index, changes the absorption properties, is the optically active molecule, many, many others. So in a simple medium, even with milk and sugar, that's two components. But if it's milk and sugar and different serums and proteins and ascorbic acid and different scattering, and the temperature goes up, therefore the scattering changes, much, much more complicated, which is why hundreds of these companies have not yet succeeded, although billions of dollars have been thrown at this problem and still are. But that's a good question. Any other questions or comments? Vandalay?
Curiosity. The bloody,
The blood?
the bloody in the redox or in they show different activity. I mean, I don't really know.
You mean hemoglobin versus oxyhemoglobin?
Yes.
Yeah. No, I don't know. I don't think they do, because it's kind of independent of that. That's more the oxygen transport, whereas glucose is more energy metabolism and stuff. So I think they're fairly independent. Actually, I should mention there's a whole bunch of other techniques simply because the problem is so interesting and important and difficult. For example, people have used fluorescence. There's a group in Maryland that has developed contact lenses that change color. So, if you want to look better and you like blue eyes or green eyes, whatever, you can actually use these fluorescently redox reaction coupled species that change color based on glucose levels in tears in your water in your eyes. The big question is, of course,
So if I drink a Coke,
well,
the color of the eyes starts to change.
that's right. If you drink more alcohol, it changes even bigger. But that's actually a very interesting question. How does the tear glucose reflect what's happening in the body? So there's a time delay and a signal change, which this group in Maryland is trying to quantify. So it's possible. There's also been tattoos, so you can do a tattoo that changes color. There's been different patches based on some of the things actually you and I discussed yesterday about interstitial fluid that's being sucked like a tobacco patch, but it's going the other way. So it's not blood glucose and it's not real time, but it's some continuous monitoring. There's been lots of work on implanted sensors. So you put something under the skin and you see if you can indwell for days, weeks, hours, months, and see how you can read out electronically or optically. All kinds of crazy things have been tried. Photoacoustics is another method. I don't know if you're familiar with it. When you shine light and you listen or you look at the acoustic signal or mechanical signal, sometimes photothermal signal as well. And there's a company in Israel that's marketing, a company called Glucon, that's marketing or it's developing and doing NIH studies on a photoacoustic method. OCT, something that I talked about yesterday, is looking at how the signal changes with depth. So not so much using OCT imaging like I talked about, but just looking at the depth profile of a single line of OCT and seeing how the slope changes. And the slope change is related to the scattering coefficient, and the scattering coefficient is related to this refractive index matching. So a whole bunch of things have been tried, and the only common thing is that none of them work, including the one I'm going to be describing, but we're trying. So how do we actually do this? Well, we need to understand the theory to understand what the signals mean to us. So we've developed a lot of kind of theoretical models of how light propagates, how polarization is lost, what effect optical activity has. And we've also developed an experimental method. The signals are so weak, the polarization retention is so low, you can't just take sunglasses, break two lenses, and look at cross-polarizers through tissue. It's not that strong of a signal. So we developed various techniques for modulating, kind of changing the polarization state and using lock-in synchronous detection to look at the polarization signals that remain kind of weak ones. We can do this in the forward direction or in the side direction, kind of rotate this all around, look in the backwards direction, etc. And again, the modeling or the theory is very important because you have to understand what the signals mean. The signals are not very simple. It's not that with no glucose you have one signal, with glucose you have a huge peak and that's easy. It's not like that. It's much more complicated. So we can define and we'll measure things like the state of the light, the optical rotation. Basically, if you come in with linearly polarized light in one plane, how much does it change? So that would be quantified by the optical rotation alpha in degrees. You can also measure how much of that Stokes vector that I mentioned has linear or circular polarization. So this would be basically the sum of the linear polarization divided by the intensity. And this would be the sum of the circular, that last element in the Stokes vector, divided by the intensity and give you a percentage. So fully polarized light would be 100%, and fully depolarized light, the one would have 1, 0, 0, 0, would be 0% for both of these. So we can quantify this retention of polarization. This is in a very simple scenario how the experiment works. It's actually not that difficult engineering-wise to engineer this. So you have laser or light source, can be a broadband light source or a single wavelength initially. You do some polarization, so make it linearly polarized, you can make it circularly polarized. Then we use a device called the Photoelastic Modulator. It's similar to, say, an acousto-optic modulator. It's basically a piece of quartz that is birefringent and vibrates, say, at 50 kilohertz or 100 kilohertz. And so what happens if you come in with linearly polarized light into this piece of quartz, say at 45 degrees, and you can imagine the 45-degree light being decomposed into a vertical component and a horizontal component. So if this thing vibrates, then the vertical and the horizontal component travel at different speeds. And so what comes out of the photoelastic modulator, depending on whether at this instant in time it's unstrained or it's stretched or it's compressed, is going to be a continuous sweep of the entire polarization ellipse. So it's going to be circular, then elliptical, then linear, then circular the other way, then elliptical the other way, then linear the other way. So what the sample sees in this configuration is all polarization states continuously changing as a function of time. And then we can use different analyzers, say quarter wave plates or linear polarizers, to look at linear and circular polarization states and measured this with a lock-in amplifier at the same frequency as this thing vibrates. And then that would be related to how much polarization is retained. So in other words, if this at that frequency measures nothing, if all the light is depolarized, no matter what you do here, then this will just measure a DC signal, and the degree of polarization is 0. So there's no information left in the polarization channels, if you want. Whereas if this measures a significant signal, then you can relate to how much polarization is retained, and you can also look at the angular dependence of this to determine if that polarization is rotated, etc. So that's the basic idea of how you do this experimentally. If you want, it's a very interesting effect called quantum confinement or low coherence. If you look in the exact backscattering direction, so exactly 180 to the beam, and you block the specular reflection, the very surface reflection, then you will always have some polarization left. In every other direction, say in the forward and the side, if this is sufficiently scattering, then eventually the degree of polarization is 0. There's no polarized light left. But there's a unique direction in the exact backscattering at 180 that will always, no matter how scattering the medium, will have some polarization retention. So there's some interest, theoretical and practical, of going exactly backscattering direction. But that's practically not easy because in that same direction as your incident beam. So you kind of block it. So you can have to play some tricks. For example, use a beam splitter and move this over here, and we've done some of that work as well. It's a very interesting effect. That's the kind of stuff that Mike Feld and that group that does sizing of particles in the exact backscattering direction, they do that.
Can't you do a kind of beating experiment?
Beating?
Yeah, because I have one polarization. When I send a strong stabilized and whatever is the polarization in this one, I can amplify or something. Because
yeah, I can do nonlinear mixing for polarization.
Yeah, you're going to beat only the same polarization.
Yeah, that's possible. I mean, this is more direct in the sense that you're actually measuring the polarization that retains in the incident beam, but you can think of better ways to do that, sure. So this is just more of electronics showing the experimental arrangement. Probably equally important, having determined and shown and demonstrated that polarization is partially retained, is what do you do with it? How do you analyze it? How do you quantify it? How do you relate it to the content of the sample? So we spent the last 3-4 years developing so-called Monte Carlo model for propagation of light in tissue that includes polarization. I don't know how much you guys do of things like diffusion theory and radiative transport theory for light propagation, and I don't know how much, if you know what Monte Carlo, does anyone know what Monte Carlo is? And yes, you do. Everyone else?
We know what Monte Carlo is.
Yeah?
Yeah, I mean, most part of the people read papers.
Right.
Does
anyone know, by the way, where the name Monte Carlo simulations come from? Why Monte Carlo?
Monte Carlo, the casino.
Yes, but why casino? Any idea?
Because you have to gamble when you're doing the model.
You're sort of right, I guess. But I mean, you're on the right track. It comes from random events when you play cards. Like in English, the game is called blackjack. 21, same game. Yes,
I
you
don't
know.
know how we call blackjack.
21, when you have 2 or 3 cards and you try to get as close to 21 without going over. Anyways, so who gambles here? Anybody? Anyways, it is very important. So what my point was that this type of Monte Carlo, there's a lot of modeling for games of chance, game theories. And what happens there is you don't know anything about a specific individual event. So depending on the cards you have and the cards that your opponent or the dealer has, almost nothing can be predicted about that particular event. But if you do that many, many, many times, you can see trends. So you know the rules of the game, you know exactly what can and cannot happen, but it still doesn't give you an idea on the individual probability, if you want. If you do that millions of times, you know that if you have a queen and a 10 and the dealer has a 5 and a 6, that you should do this or not that in many, many times. So the same idea here. We know exactly how light propagates in single scattering events and single absorption events and single optical rotation, single retardation. What happens if it happens billions of times? You can't model that properly using electromagnetic theory or Maxwell's equations. You don't know the boundary conditions for all these optical inhomogeneities. Even if you did, it'll take you years to solve it because it's not just planar interfaces. So the approach is to use this single events whose physics you know and understand very well, and just repeat it many, many times in the computer, basically gamble in the computer using a Monte Carlo approach. So play it again and again and again, and after, say, 10 billion photons, quantify how much comes out in this direction, or how much polarization is retained as a function of the properties of the sample. So this Monte Carlo approach was developed actually in Steve Jacques' group and Li-Hong Wang's group for biomedical optics. In fact, that code is now freeware, shareware, anyone can download it. That was in the late 80s, early 90s. And then in the last 10 years or so, a lot of people, including us, have been trying to graft on or to add to that intensity distribution, Monte Carlo, the Stokes vectors and the Muller Matrix analysis.
What is the address we can download?
The what?
Where is available?
I don't remember the website, but it's quite easy. If you look at MCML, which is the Monte Carlo multi-layer, it's a freeware, free of charge, available from Li-Hong Wang and Steve Jacques. Those are the two authors, probably 1989, and there's new versions, 93, 94. If you guys can't find it, I can go back on the web and find it.
What is the main difference of light in tissue and light in random disordered medium?
Very little difference. I mean, it's the same.
This is back on the 60s when people...
Yeah, planetary atmospheres and remote sensing. Yeah. So this is specifically tailored for tissues. It looks at things like scattering anisotropy, the G factor, the effect of the mean cosine of the scattering angle. It's adopted somewhat to the range of optical properties that you would find in tissue, say a scattering coefficient of 100 inverse centimeters, a G of 0.9, absorption coefficient of 1 inverse centimeter. So it's not, it's just something that's very applicable to biomedical optics, let's put it that way. So our approach is to go the same route, but
still it's a program without large scale structure, right? Or they can include everything. It have a lot of layers.
Yeah. Oh, yeah. You can do that. Absolutely. That's why it's called MCML. So Monte Carlo multi-layer. So it's a layer geometry. However, for example, in our lab, on the work I'll talk about tomorrow, we're doing more interstitial. So we don't need layers. So we had to adopt that to a different geometry. But you can do that and put in interfaces. No, it's for sure it's not a random homogeneous media. It's random inhomogeneous media with layers and structures, and you can model the eye, or you can model the skin with different optical properties of the skin, etc. So anyways, the Monte Carlo is being adopted for polarized light in this context, and this is just a graphical representation. So I kind of described that. So what you do is you track many of these individual photons. You know the probability distribution for each one, but you still can predict what's actually going to happen when you shine a billion of them. So you repeat this many times, typically 10 to the 8th. That's for intensity. If you want polarization effects, which is what we want, quite often this number goes up to 10 to the 9th and 10 to the 10th. Again, if you have very low absorption and very high scattering, then the number is much higher because the photons travel a lot. If you have very high absorption, they're very quickly cease to exist, they extinguish, and then you can get away with average properties, say for 1 million photons. So you record the resulting statistics, you calculate cumulative properties, and you average them to yield something that you can experimentally observe and quantify. And the big thing is, how do you add polarization to this whole business? And so there's quite difficult mathematics based on that Muller Matrix approach that I described briefly to do that. So again, calculate macroscopic or experimentally observable properties from individual interactions. You know the individual physics, but what's going to happen in a crowd of these photons when it actually interacts with the real medium? So this is a little more. using this Monte Carlo gambling approach, and you can quantify and actually form images of different, so this is intensity, this is the circular, the two linear polarization states, this is circular polarization states for incident unpolarized light, incident linearly polarized light in this plane, incident linearly polarized light in this plane, and circular incident polarized light. And of course, you can now see, for example, if the medium that you're modeling has glucose or optical activity, how does this change? Or if the absorption changes, or if the scattering changes, or if the scattering stays the same, but the anisotropy, the G factor, the average cosine of the scattering angle changes. All of these things have an effect. So in the model, you can actually do that if you trust your model. So we spent a lot of time developing this model and validating, because there's so much computer stuff. If you put in, garbage will come out. So garbage in, garbage out. So we spent a lot of time experimentally validating and comparing with other people's results, comparing with some of the planetary atmosphere early research to make sure we're on the right track. So one other thing that I didn't mention before is that it also allows us not only to calculate the polarization properties, such as diffuse reflectance in any direction, but also allows us to quantify the average path length. So when we shine light through a clear medium, the path length is just the thickness of the medium. If this is now milk and I shine light and I detect it 45 degrees or 90 degrees, what am I detecting? What have these photons sampled? Where have they gone? So we can actually quantify where they go and what is the average path length, or the statistically average path length. And that gives us an idea of how we can extract the optical activity and how we can relate that to glucose levels. So this is just a bit of an animation. This is showing an actual image of some of the Stokes vectors. So we have incident, so it's a cube of material, so not a very good model for a finger or biological media, but it's a start. It's a start. So we have incident pencil beam coming in from the bottom, and this is just showing the intensity where the light is distributed in 3 dimensions. And this is the property, so this is fairly heavy glucose concentration, fairly low, relatively speaking, scattering coefficient. And this is looking at the linearly polarized light component, and then we can compare this with, for example, 0 glucose and try to quantify differences. So this is just giving a graphical representation of what these results can look like.
Sorry, Alex.
Yep. Don't be sorry.
In this intensity part of the vector.
Yeah.
It seems interesting that you have a lot of light coming at the face that is 45 degrees.
You mean 90 degrees?
Back.
90 degrees?
90.
You mean 90 degrees?
90 degrees, sorry.
Yes, yeah.
And then you should have also a lot of light coming in the direction
In the back?
on the back.
Yeah, no, actually, you do. This is a little.
You are looking at the 90 degrees probably.
No, we're looking everywhere. It's just we're not scoring. You're right. In this particular orientation, we're not scoring because we want to show where the incident light is. But absolutely. Mind you, right at 180, the reflection is not very large. There is a fair amount of light, almost this level around here. We're just not showing it. Yeah, retroreflection is right here. So generally not very.
So I'm not worried about the retroreflection or the scattering light, because if you are scattering that amount, at 90 degrees,
there's going to be
you
some
probably
in
have
120, 130, absolutely, yeah. So this is just for display purposes, but also wanted to get back to this retroreflection. It's actually not very intense. It's just very unusual because of this weak localization that it retains polarization to a much greater degree than just about any other direction. But that's a good point. There's light at the bottom as well. This is just, again, a simple graphical representation of what glucose can do. So this is a side view, square medium in depth this way. So kind of like an optical cuvette lying sideways, incident light, 90-degree detection, and we're modeling what's happening as we add more and more glucose. This is significant addition of glucose, and it's changing the refractive index. So that does 2 things. It lowers the scattering coefficient, and it makes the scattering much more forward peaked. So as opposed to being more side scattered on individual scattering events, it's now more forward peaked. And so you...
Just to,
Absolutely. No problem.
To understand what we're looking, we are only looking the with photons that start here and end here. Right?
Absolutely. There's many others we're not scoring. So we're only interested in this case, what are we detecting? If our detector was 90 degrees, this is not even polarized light or not polarization. We're just tracking the path length to see where's the light going. Is it sampling here? Is it sampling here? If we detect something at 90, what are we actually looking for in tissue? Is there structure in tissue?
So over here, on this place, you have like a single point.
Yeah, incident beam.
But you're modeling like a window on the other face, right?
Well, that depends on what we say is the,
size of the
is the solid angle of the detector, the viewing. Depends on the solid angle of the detector and the projected area. So if there's a lens here, we would be modeling a much smaller point. If this is a broad area detector or several millimeters, that's very close, we'll be modeling a much larger point. Yeah, I mean, that's one of the advantages of having a legitimate validated model, is you can model experiments to understand the data, but you can also predict what you should do.
Then you can do that on yourself.
Right, so instead of running these endless experiments and not knowing what's going on, you can use the model to guide the next set of experiments. You should not go 90 degrees. You should go to 175, and we've done a lot of that. And you should look at a much smaller detection area versus a much larger detection area, or vice versa, depending. So the model is very important to analyze the data you got, but also to plan the next set of experiments.
And not waste too much time.
Yes, although the Monte Carlo is not very quick with that many photons. But there are ways with computing powers to set up many computers, run it over the weekend, come back Monday, and know what to do. Absolutely. All of these things are, I mean, the Monte Carlo doesn't have wavelength in it, but you have to put in properties of the media. And the properties of the media are different in near infrared versus they are in the visible. So it is implicitly depending on the wavelength, of course. Yeah. So this kind of approach, as I was saying, gives us a quantifiable predictions. We can detect how much light intensity-wise is going to be for different geometries, different optical properties. The path length, we can quantify and relate this path length to the retention of polarization, to optical rotation, and relate all those things to glucose in an ideal world. You've seen those rotating cubes before, so you can just ignore them for now. Just highlighting some of the experimental parameters that we've determined, such as the polarization modulation to increase the signal-to-noise ratio. We quite often use balance detection. Those of you in electronics know that you can reject the common mode noise, the common mode rejection ratio, for example, if there's intensity fluctuations in the light, which are unrelated to your sample properties, but will show up as fluctuations in your signal. I'll talk about this result a little later on, I think, in 1 or 2 slides. So these are some of the our relatively early results, looking at degree of linear polarization, so that thing that I call beta L. So another slide. As a function of glucose concentration, this is very heavy glucose concentration. The physiological range is basically here. Fairly tissue-like scattering, but maybe not so much. 30 inverse centimeters is kind of semi-transparent tissue as opposed to really turbid tissue, so this is early results. And this is looking, so the retention of polarization and rotation of linear polarization state. And you can see that we're kind of approaching the physiological range, and we can tell the difference between clear media and scattering media, but there's a lot of work to be done. And again, to go back to a point I mentioned before, this is a very simple system. So this is scattering, which we can control, and various amounts of sugar or glucose. By the time we get to real tissue, it's going to be much more complex. So we can measure depolarization. We have this Monte Carlo theory to relate this to actual quantifiable sample properties. And our plan, which we haven't done, is to actually do this at several wavelengths, because there's no hope of being able to use a single color light in a real tissue, given the complexity and the compositional problems in tissue. Here's another experiment, a very simple one. So what actually happens, so this is degree of linear polarization, everything else the same. This is scattering coefficient, basically, or the concentration of scattering particles. We can convert that quite easily to a scattering coefficient. So this is no sugar, so basically just plain milk if you want. And this is fairly sweet milk, about 100 times that of physiological.
So what is actually your sample?
Typically,
Control that. You're not working with tissue right now.
we have done some tissue work, but typically we have polystyrene microspheres of the size of half a micron to 1 micron. And from first principles, from something called MIE theory, M-I-E, we can easily calculate the scattering coefficient at a given wavelength in a given refractive index medium. So we want, based on our Monte Carlo, we'd like to have 100 inverse centimeters with a scattering anisotropy of 0.8 or 0.9. So we do that, and then we make a sample in water with a known amount of scatterer. And then we want to vary, for example, the glucose concentration. So we add various amounts of glucose, and that would be our first simple sample.
And then, how do you know the amount of glucose?
How do I know the amount of glucose?
How do you know that the amount of glucose is not causing different aggregation stage?
Well, glucose dissolves. Glucose dissolves in
but
water.
The molecule sticks to the surfaces of the spheres.
It may not do that. I don't think it does. There's no evidence that there's surfactant or surface absorption.
Okay, so that's the answer.
Yeah.
It doesn't do anything.
I'm not a surface chemist, so it may do something, but it's not.
They use a polyester sphere, microspheres, to study phase transition in those solutions.
Of glucose?
No,
phase transition. Yeah.
Based on.
Yeah. I don't know. We have a surface chemist, so maybe you can tell us if glucose adsorbs onto polystyrene or not.
Or causes different. Probably not.
Yeah. I don't have all the answers, right?
Forget about it.
Okay. So anyways, so here is the retention of polarization. So this was one of our early results, and very encouraging one at that, in that it indicates that there is measurable signals, and they differ for glucose versus non-glucose. But it's not clear whether this is to do with the optical activity of glucose, or more likely just due to the refractive index matching. Simply because this just has lower scattering coefficient. Because we've added so much glucose, this becomes transparent. In fact, in science fairs, or in, as you say, diffusion, it's a pretty interesting experiment. If you have milk and you keep adding sugar to it, and you add a lot of sugar, dissolve it, eventually it becomes transparent. So a glass of milk can begin to look like this.
You can see also the beam bending.
Right. That's easier to do with glycerol, actually, not with sugar. But again, so if you have a glass of milk and you add enough sugar to it, like really, really sweet, and you dissolve it because it takes a lot of stirring, you can get the glass of milk to look like this, to look like transparent water. So that's what's probably happening here, is just refractive index matching. But so we can have significant polarization preserved up to optical depth. Optical depth is the product of the scattering coefficient and the physical depth, up to about 15, which is fairly optically thick. That's how you can quite often quantify, thickness of clouds and so on. The presence of glucose increases the linear and circular polarization retention, and there's a lot of controversy in the literature whether this is due just simply to refractive index matching or to something called circular polarization lines, eigenmodes. Circularly polarized light is actually better preserved in tissue than linearly polarized light, at least in the forward direction, and this has to do with scattering anisotropy, and there are several papers, theoretical papers, addressing and measuring that point, and we've seen that before. So degree of circular polarization is actually higher than degree of linear polarization. And if you have glucose in the medium or any optically active materials, the light decomposes into circularly polarized eigenmodes when it travels through that medium. So there's some theoretical reasons to believe that circularly polarized light should be better preserved in tissue, which is what we've seen, and even more so if you add glucose or if you have higher glucose in tissue. But that's to be seen. So we've also tried to see if optical activity makes any difference. So glucose, as I said, is chiral molecule. So for every right-hand glucose, there's a left-handed glucose. Although in biological tissue, all of glucose is left-handed. There is no right-handed in any animal or human or anything like that. So you can actually get from a chemical company.
Why?
There's a lot of evolutionary reasons, evolution, and actually very interesting.
It's the chiral problem, right?
Chiral, yeah.
Chiral.
Yes, yeah, yeah. So it's just left-handed glucose is what we all use and like, and right-handed glucose is complete.
Life would be different.
Yeah.
To us, like.
Supposedly, that there's a lot of kind of philosophical implications to that. And actually, if you know, there's a terrible disease or a terrible, I don't know if it happened in Brazil, in North America, in Europe, about 40 years, 50 years ago, there used to be a medication that's given to pregnant women, Thalidomide. I don't know if you've heard of it. Apparently that's an excellent drug, and the reason it had such devastating consequences was that the chirality was not controlled. So it was the wrong isomer, the wrong form of thalidomide that caused all these terrible birth defects. Yeah, short limbs. And so now actually that drug is being resurrected, although it's got a terrible image because people are afraid of it, but it's being done right, and when it's put in the right, in its correct chiral form, the right isomer, it's actually working relatively well, both for pregnant women and as a chemotherapy drug in cancer. So a little difference can make a big difference. So anyways, to get back to what I was doing, we decided to form mixtures of left and right isomers to see if we can still see the difference in turbid media. And we could have done this with glucose, but the right isomer of glucose is extremely expensive because it doesn't really exist. So you could make it in a laboratory, but it has to be by artificial methods. You cannot use any life forms to make it because no life forms can live with it. So we just chose another sugar from a chemical company, something called arabinose, which is similar to glucose, which can be in left and right forms quite easily. So this is showing basically refractive index for something that's optically active, so one form of arabinose, something that's optically inactive, which is glycerol, another method of changing refractive index, and something that's optically active but in the opposite sense. So a mixture of left and right, or so-called L and D. And we see that actually the chirality makes no difference. So this is circular polarization retention, and this is linear polarization retention. So it really implies that what we're measuring is just refractive index matching. It doesn't matter that the glucose happens to be optically active in this case or not, at least for polarization retention. And this, of course, is dependent on the tissue properties, etc. And this is the slide I was showing you before. So this is the physiological range here. This is optical activity or optical rotation as a function of glucose concentration. And the slope of this line very much depends on the scattering properties of the tissue. That's to be expected. This is an interesting experiment we did also. We wanted to see if we can determine the handedness, the left versus right states, again, in a glass of milk or in a scattering medium like polystyrene spheres. So we can actually detect the difference between a left form of an isomer and a right form of an isomer. Again, this is trivial in transparent media, but quite tricky in scattering or in turbid, cloudy media. Although there is an offset, you notice it doesn't start from 0, which we don't quite understand. It should start from 0, but it's starting from 1 degree offset. Any questions, comments?
So what is the difference between this experiment and the other one that you saw no effect?
Well, there this is actually measuring optical rotation. The other was just measuring the retention of how much light is polarized, so degree of polarization, without saying if it was rotated or not, how much it is retained. And the retention seemed to be just as a function of refractive index. It had nothing to do that this is handed or not handed. This is showing that we can actually detect molecular symmetry in diffusive scattering, although, as I said, we don't understand why this is offset to 1 degree. Probably to do with scattering as well. So we've developed some methods of quantifying path length and how to relate the actual. So we measure 2 degree optical rotation. Well, that's nice, and we can publish that, etc., and quantify it, but what does it mean? How much glucose is actually present? Can we relate that measurement to the concentration of sugar in our sample? So for that, we need some model. We need to know how much light has traveled, what is the path length, and can we use a simple relationship such as that the measured rotation is related to its concentration and to the path length, and this is the proportionality constant. This is like a physical constant for glucose, the optical rotatory power, which is wavelength dependent, temperature dependent, etc. So that certainly works for clear media, and that's how it's used to quantify sugar concentration in the clear apple juice, for example. Does it work in turbid media? And in turbid media, we don't know this, but we can get it from models. For example, we can measure or model the degree of linear polarization and try to relate that from our model to the path length. And then maybe from the path length, get this concentration from the measurable this and measurable that. And we can see if this approach works by adding different amount of absorbers to our scattering medium, because the absorbers will basically make the path shorter and shorter. It'll decrease the amount of light that we can detect, but it will increase the polarization that we detect, because the multiply scattered light is not going to make it to the detector, because the absorption, if there is absorption present, is going to negate that. So one way to increase the degree of polarization is to add absorption. You decrease the amount of light, but whatever light you do detect is more polarized. So for example, if we did some skin experiments and people like pigmented with black or negroid skin have much less light coming back, but the degree of polarization of that light is much higher. Whereas white people or North Europeans have much more light coming back, but it's mostly depolarized. So it's kind of opposite to what you expect in some ways. So we did some modeling and some measurements and developed kind of an algorithm of getting the degree of linear polarization, which we measure experimentally, the optical rotation. Then through our Monte Carlo model, get the path length, and that's indicated on this graph. And then either through model or this equation again, try to get the glucose concentration from this path length and from the experimentally measured optical rotation. It kind of works in principle in simple systems, but getting back to a question we had earlier, what's going to happen in tissue? So glucose is not the only optically active species. Scattering can change many ways depending on temperature, depending on physiology, depending on pigmentation, etc. So the idea, which we haven't implemented yet, but we have some grants to do that, is to do this as a function of wavelength. So basically do polarization spectroscopy in turbid media and see if we can tease out some of the things we've learned at single wavelength and quantification, now put in a more encompassing wavelength dependent model and basically do polarization spectroscopy in tissue. Okay, so that's all I wanted to say about the glucose story. Took a little while, but hopefully you kind of understood where I'm going with it and stuff. So it's work in progress. We've done some clinical or preclinical volunteer experiments. We haven't looked at diabetic patients because we're not ready for that yet. And it's, again, a very complicated problem because, you know, I go eat a chocolate bar now. Is which polarization properties of the light that's going to be detected are going to be quantifiable and related to what's happening to that chocolate bar, and how does it differ from me to somebody else who has different metabolism, different skin pigmentation, different level of, you know, surface sweat, et cetera, et cetera. Many, many variables. So we're hoping to use the wavelength properties of the polarized light as opposed to just wavelength properties of the diffuse reflectance to do everything that diffuse reflectance can do, because we can still measure the intensity as a function of light, but now we have all this polarization information and some polarization knowledge of how to interpret those signals.
So the idea is to do a wavelength dependence.
Polarization.
For sure you're going to find out what are two wavelengths that are very relevant.
No.
You can do ratio? And
yeah, I don't think so. I don't think it's going to be as simple as a couple of wavelength ratios, you know, hemoglobin, oxyhemoglobin. It's not going to be that simple. So we're going to probably use the whole wavelength range from, say, 6 or 700 nanometers to 1450.
Real spectroscopy.
Real spectroscopy, yes, the real thing. Right. So given the fact that we've developed all this platform, both experimental and theoretical, for understanding and modeling and measuring polarized light and multiply scattering turbid media, another very interesting biomedical applications came along, which was very appropriate for this method, for this methodology. And this is using this birefringence, quantification of tissue anisotropy, for birefringence. Specifically, we have a large program in our hospital, so-called regenerative medicine. So stem cells to grow back tissues after surgery, after some kind of a heart attack, after radiation therapy, when there's normal tissue damage inadvertently. So, specifically in the heart, we have a very big program in cardiology in our hospital with stem cells, etc. So the colleagues are very interested in quantifying collagen birefringence following stem cell therapy. And following injection of stem cells after a person, in this case, has a heart attack.
See?
No? Okay. Good. Thanks, Manuel. So the idea is, can we tell the difference between normal myocardium, so normal muscle of the heart, versus, there's actually another panel I should have put in here, and that's infarcted myocardium. So this is following a heart attack. So normal following a heart attack, and regenerative. So the one that grows back following normal scar tissue formation or following stem cell therapy. Can we quantify those differences? So can we use polarized light as a non-invasive probe for quantification of birefringence, tissue anisotropy? I've covered some of this before. What practically happens is birefringence basically acts as a wave plate or as a retarder. So it changes, it does not depolarize the light per se, but it just changes the polarization state. So if you have a transparent medium, for example, a piece of plastic, and you stretch it and you shine in linearly circularly polarized light, you still get polarized light out, but the polarization state changes. The polarization ellipse is altered, which is how, for example, in engineering, you can look at weak and strong points of a bridge. If you put it between cross polarizers, where the structures are more birefringent, there's more strain and more stress, you can see different colors, different polarization states. So light is still polarized, but the polarization state has changed. So what's happening practically is the anisotropy of tissue transfers the linear polarization to elliptical or to circular. So if we come in with this, we'll come out with that, roughly speaking, in an ideal world. And we can quantify how much that's happening and relate that to tissue birefringence and anisotropy of collagen. So this is some more structures, different types of collagens, according to the literature, according to polarized light microscopy, display different anisotropies, different organizations, short order and long order. And so we're going to investigate the potential of polarized light to use this for tissue characterization and assessment. This is just some early experiments. The experimental setup was actually quite simple, at least at a single wavelength. So as I mentioned before, the experimental setup is not too different from what we had before. What's different is the model. The model becomes much more complex because modeling birefringence or tissue anisotropy, so direction-dependent changes in tissue properties, is very difficult. That Muller Matrix that I was mentioning, the 4x4, therefore 16 elements, is a real nightmare. Very complicated. Direction and the amplitude of the retardance, and you're modeling real tissue, so you have scattering, there's birefringence, and you still have optical activity, whether you're interested in it or not. For glucose, it's still there. So this is back to this problem where the matrices so-called don't commute. So you can't have A times B because it's different from B times A, and both are wrong because A and B are happening at the same time. So there is a very relatively sophisticated approach called an N-matrix approach that we've adopted from clear media that's been developed maybe 15 years ago for exactly the same models for trying to model two simultaneous effects, basically using differential. So instead of going A times B, you go dA times dB, a little differential in A and B, and then you integrate exponentially over the two, or over 3 or over 4, depending on how many effects you have. The problem to do this in scattering medium is very difficult. So it took one graduate student a good 3 years to develop a model and to validate the model experimentally to do this properly. So we have some results and it looks pretty good, and so we can investigate this ability to quantify birefringence. So these are some of the early results. So we're measuring, again, this is not too different from the microsphere experiments, except in addition to microspheres, this is now made into a solid sample, and we control the birefringence. So we stretch it. So it's kind of a gel, and then we stretch it to a given extent, and we can, in the absence of microspheres, we can stretch it and quantify the retardance as a function of stretch. And then we have microspheres. The situation becomes more complicated, but since we know the stretch and how it relates to retardance, we can form these types of graphs. So this is coming in with circularly polarized light, looking in the forward direction and looking in the 90-degree direction. Actually, 90 degrees actually this way in this geometry. And looking as a function of extension, or retardation, or birefringence, if you want, on the vertical axis, how the incident circularly polarized light is lost and how the linearly polarized light is increased. So it's really acting as a quarter wave plate or a wave plate. And the sum of these two will give us the degree of total polarization, which is actually going down because there's scattering involved. So it gives us a whole bunch of information. And we can do this in the bottom direct. So this is forward direction. And this is side direction. And just to quantify the same graphs as what you saw before. So this is actually our model, is the solid line, and the green line is the measurements. So the match is pretty good. The agreement is fairly good. If you add this as a function of extension or this as a function of extension, you see that the total degree of polarization is going down because of the scattering of the sample. And you can actually see that it changes from input circular to output linear polarization. One is going up, which didn't exist before, and the one that's incident is going down. And there's slight differences as to whether this occurs in the forward or in the lateral direction. So we're quantifying that as a function of input parameters. We've done very limited ex vivo real tissue experiments. So this is showing basically a real biological sample. This is not polystyrene spheres, but actually heart, a thin section of the myocardium. Sorry? Ex vivo, correct. Yeah, we haven't done in vivo this.
So is that after mutation, just taking stem cells for a long time?
It's not a patient, it's a chicken. It's, sorry. So
Okay,
no stem cells in this chicken, no, not yet. But actually, the group that we work with has a lot of animal models for animal regeneration, like rats and mice. They're very, very good at this. So when we're ready, we'll do this properly, not just on chicken hearts. So this is looking at, as a function of tissue thickness and as a function of stretching as well, what is the retardation doing? So we do have some ex vivo real tissue data. What we've also gone is in vivo, we're not going to be measuring through thin sections in the forward direction. We're going to be measuring in the backwards direction, most likely. Not necessarily at 180, but somewhere in the backward sphere. So we've done a lot of simulations and some initial experiments in the reflectance geometry. And one of the nice things we found is that the results we're getting are almost independent of the tissue properties, which in general we don't know. We don't exactly know if the scattering coefficient of this heart is going to be 150 inverse centimeters or 100 or 85, or the scattering anisotropy or absorption. So this is showing the results or the retardance as a function of the difference in refractive index for different optical properties. And the results are not too sensitive, which is a good thing for us because we don't know the true optical properties in an in vivo context. So this may be a little more practical for eventual applications. So I probably talked long enough. Let me just conclude, and maybe we'll have some questions if you guys have some. So in the grand scheme of things, what does this all mean? So probably the biggest thing is that we have a theoretical experimental method to measure and quantify the retention of polarized light in multiply scattering tissue media or tissue-like media.
So just a question. Do you put your sample on like a little micrometer holding device?
To stretch? Yes, yeah, for that one.
So you can quantify actually how much?
Absolutely, yeah. So the whole approach of our laboratories, we do a lot of clinical, a lot of experiments, but we have to quantify. We have to understand the numbers.
This tissue, after you take it from the
That we don't.
That animal
Yes, we
is
cannot.
gonna, dehydrate.
Yeah.
How do you control it?
We add serum, or we add physiological saline to keep it moist. But our idea, of course, is to go in vivo and eventually through optical fibers. And then we're not going to be, but we haven't done that yet. So for now, we just keep it moist with saline, physiological, like with water, almost with water. So again, when I was mentioning, we have a platform and a method of experimental and theoretical measurements, experimental measurements and theoretical quantification, so polarized light in tissue or in tissue-like media. We can measure some properties such as the optical rotation, linear polarization states. There is some potential for determining the path length and glucose concentration, although that's a big question right now, along with everyone else's big question. We can starting to use this to quantify the anisotropy of tissues. The heart is just one example. This is not the only one. We have a platform both for analyzing the results we get, but essentially even more importantly for predicting what we should do. What is going to be the effective wavelength, or what is the wavelength range that we should set up our experiments for, and should we be measuring in the forward direction and the side direction at 150 degrees, 180 degrees, etc. And so I guess it's a bit of an interesting novel method for examining turbid tissue with polarized light, with, at least for my mind, these relatively important applications in red, so glucose and Birefringence. And I think that's just another summary. So we can use this to quantify optimal geometry. And as mentioned before, how do we account for this glucose unrelated signals, glucose unrelated optical activity analytes? We're developing this into spectroscopy, into tissue or turbid polarimetry, doing this in myocardial birefringence. So the idea with spectroscopy is we're going to get the same information in the intensity part of our signal as what people do that do absorption spectroscopy, and they're getting pretty close to glucose determination. So if we now add the polarized light information and analyze this with partial least squares and chemometric signal analysis, is that enough to get sufficient accuracy, specificity, sensitivity? And we don't know the answer to that question. And we have further studies planned. And that is my last slide. So thanks very much for your attention. So questions? No?
I can start.
Sorry?
I can start with
Yeah, please start.
when you do the Monte Carlo,
Yeah.
you find the measurement and the Monte Carlo result like good agreement.
Sometimes, yeah. In some,
Decent agreement.
yeah.
Do you have any empirical relation for that dependence? I mean,
Somewhat.
empirical relation. Can you come with a function? So
Yeah.
go back to
I know, I understand, sure. Yeah, it's easy. I mean, you do a polynomial fit and you say, well, this varies as an exponential and the path length decreases exponentially.
This is exponential.
No, oh, you mean for that particular one? Yeah. I'm not a big fan of empirical relations. I think if it helps you in further predictions and modeling, that's a great thing. But we have a much better way. I mean, it's longer because we have to run Monte Carlo.
Yeah, but the question is if you're going to do things in real time sometimes.
Yeah, sure.
Then
Yeah.
doing Monte Carlo,
Yeah.
measure and wait until tomorrow morning.
No, absolutely. So, in the absence of any,
You're not in that phase yet, right?
in the absence of any information, of course, it's easy to say that the polarization is going to be exponentially lost as a function of optical thickness. Right. And you can see the graphs, and it kind of makes sense. But yeah, I don't believe in those things in the sense that that's a very general statement. So the rate of polarization loss is going to be very much dependent on.
Yeah, but I'm not.
No, I understand.
What
Yeah.
I'm saying is you do the Monte Carlo.
Yeah, and you can
And
see
you
the
know-
trends. Yeah,
You know
yeah.
you don't know why, but you can find
It seems like it varies as 1.5 power. Absolutely. Yeah,
that can be used for
prediction.
prediction.
Yeah, no, that's a good point. Yeah,
This is what we do for trying to find out the distribution of light.
yeah,
The distribution for PDT.
sure.
Because if you do the Monte Carlo, which
Yeah, no, but for distribution of light, you can use diffusion theory. That's analytical, and that's
when you start to put all the
Well, if you're developing a commercial device, I'm taking a point of view of patient.
yeah.
If you're developing a commercial device, that's supposed to take seconds.
Sure, no, but I mean, even Monte Carlo, you
But
can
that's going to
create.
vary with your light temperature.
Yeah, no, but you're right. But
It's
even
gonna
in
vary
Monte
with-
Carlo
so
ventilate,
if
you can
have
create.
all these empirical